Hey everyone, and welcome back to the Deep Dive. Today we're going to be talking about something pretty wild.
Yeah.
These things called acellular pathogens.
It's a mouthful.
It is, and we've got some really fascinating excerpts to help guide us today from an OpenStax microbiology textbook. So
Excellent source.
really good stuff. I guess this listeners, these things cause disease, but they aren't even technically alive.
It really makes you think, what does it even mean to be alive? What's the definition?
Right. Totally does.
Yeah.
So let's start with probably the most famous cellular pathogen viruses. I mean, we hear about them all the time. But here's something that might surprise you. They don't have the stuff they need to be considered alive.
Right.
Like no ribosomes, no metabolic processes.
None of the things you'd normally see in a living thing.
It's true.
Like if you were to look at a cell under a microscope, you'd see all these complex structures working together,
Yeah.
But viruses, they're like these minimalist invaders, just genetic material.
And that's either DNA or RNA.
Right. But never both.
Never both. Interesting.
All tucked away inside a protein coat.
It's called a capsid.
Exactly. The capsid.
And to replicate, they basically hijack a living cell.
Yeah, they just take over completely and turn it into a virus factory.
It's like they have the blueprints, but they need a construction crew to actually build anything.
Perfect analogy.
And they force the cell to become that crew.
Yep. And some viruses are even sneakier.
Oh.
Yeah, some of them actually steal an extra layer from the host cell.
No way.
As they're leaving, they're like, oh, and I'll take this too.
Oh my gosh, so rude.
It's called an envelope.
An envelope, okay.
Yeah, and it basically helps them disguise themselves even better, blend in.
Wow, so sneaky. Like, thanks for the ride in the coat?
Exactly. And get this, viruses, they come in all shapes and sizes too.
I imagine so.
Yeah, that actually helps scientists classify them.
Okay, so how do they do that?
Well, they look at the shape of the capsid. It could be helical, like a spiral staircase, or polyhedral, like a 20-sided die, or sometimes it's even more complex than that.
Wow.
Yeah. They also look at what kind of genetic material is inside, like is it DNA or RNA?
Right.
And importantly, what kind of cells the virus infects.
Oh, that makes sense.
Yeah, because some viruses are generalists. They can infect a wide range of hosts, like, different animals or even plants. But some viruses are super specific.
Oh, really?
Yeah, they might only be able to infect one particular type of cell in one specific organism.
Like a lock and key kind of thing.
Exactly.
So you've got these incredibly tiny things that we can't even see without a microscope, and they're wreaking havoc.
Causing all sorts of trouble.
How do scientists even study them?
It takes a lot of ingenuity for sure.
I bet.
Yeah. One of the big challenges is that to replicate viruses need a host cell.
Okay.
So imagine you're trying to study a virus that infects bacteria.
Okay.
Those are called bacteriophages, by the way.
Bacteriophages. Okay, I've heard of those.
To grow them in the lab, scientists basically create this bacterial lawn.
A bacterial lawn, what is that?
So picture a petri dish, the round dish they use in labs, and they spread a layer of bacteria all over it.
So it's literally like a lawn, but made of bacteria?
Pretty much, yeah.
That's kind of disturbing, but also kind of cool.
Right. And then they introduce the bacteriophages onto this lawn.
Okay. And then what happens?
Well, as the bacteriophages infect the bacteria, they start replicating and they create these clearings in the lawn.
Oh, so it's like they're mowing the bacterial lawn.
Exactly. And each clearing is called a plaque.
A plaque.
And each plaque represents a single viral infection, so it allows scientists to isolate and study specific viruses.
Oh, that's so clever.
Yeah. But there are other ways to study viruses, too.
Oh.
Molecular methods like PCR, for example, that can amplify viral DNA or RNA, which makes it much easier to study.
And what's PCR again?
It stands for polymerase chain reaction.
Right.
It's basically a way to make millions of copies of a specific DNA or RNA sequence.
Okay, got it.
And scientists also look for the damage that viruses cause to cells.
Okay, like what kind of damage?
Well, there are a lot of different things. It could be changes in cell shape, or the cell might start producing weird proteins, or it could even die.
Oh, wow.
Yeah, these changes are called cytopathic effects.
Cytopathic effects.
And then there are also immunological assays that detect specific viral proteins.
Okay, so it's like a detective story at the microscopic level.
That's a great way to put it.
So let's say a virus has managed to land on its target cell.
Okay.
What happens next? Like, how does it actually get in and cause all this trouble?
It's a fascinating process, actually. It's called the viral life cycle.
The viral life cycle? Okay, break it down for us.
It starts with attachment.
Attachment.
The virus has to attach to the host cell.
Okay.
And it does that by binding to specific receptors on the cell surface.
So it's kind of like finding the right key for a lock.
Exactly. If the virus doesn't have the right key, it can't get in.
So the cell has some defenses then.
Oh, yeah, for sure.
Yeah.
But viruses are constantly evolving to try and overcome those defenses.
That's pretty scary.
It is.
So the virus is attached. Now what?
Now comes penetration.
Penetration, so it's getting inside.
Yeah, and there are different ways this can happen.
Oh.
Bacteriophages, for example, those viruses that infect bacteria, they just inject their DNA directly into the cell.
Oh, wow.
But animal viruses have other tricks up their sleeves.
Like what?
Well, some of them fuse their membrane with the cell membrane.
So they basically just sneak in?
Like undercover agents, yeah.
Wow.
Or sometimes they trick the cell into engulfing them.
It's like that Trojan horse story.
Exactly. That process is called endocytosis.
Oh, right. Endocytosis. I remember learning about that.
Yeah, the cell thinks it's taking in something useful, but it's actually letting the enemy in.
So sneaky.
And then once the virus is inside, it's time for uncoating.
Uncoating, what's that?
The virus basically sheds its protein coat and releases its genetic material into the cell.
So now the cell is really in trouble.
Yeah, things are about to get a lot worse for the cell.
Okay, I'm scared for the cell.
The next stage is biosynthesis.
Biosynthesis.
This is where the viral genes take over the cell's machinery.
And what happens then?
The cell is forced to start producing viral proteins and making copies of the virus's genetic material.
So it's basically become a virus factory.
You got it.
Poor cell.
And once all the new viral parts are made, it's time for maturation.
Maturation.
This is where all those new viruses assemble.
Okay.
They package their genetic material inside new protein coats getting ready to go.
Like little virus babies all dressed up.
Kind of, yeah.
And then what's the last step?
Release.
Release, so they escape.
Exactly. The new viruses burst out of the cell.
Wow.
Often killing the cell in the process.
Oh, it's sad.
It is. But it's the way of the virus. You know, they have to spread and infect other cells.
That's like a hostile takeover. The cell never stood a chance.
Nope, not really.
But here's something I always found mind-blowing. Some viruses can actually hide inside a cell.
Yes. That's called viral latency.
Viral latency, okay.
Instead of immediately replicating and bursting out, some viruses can integrate their DNA into the host cell's genome.
What?
Yeah, and then they just lie dormant.
For how long?
It could be days, months, even years.
That's crazy. It's like a ticking time bomb.
Yeah. You might think you're in the clear, but the virus is just biding its time, waiting for the right moment to reactivate.
Oh, man, that's terrifying. So it's like a sleeper agent?
Exactly.
I've heard that this is what makes diseases like herpes so hard to treat.
That's right. The virus can reactivate when the host's immune system is weakened or under stress.
Oh, that's no good.
Leading to a recurrence of the infection.
Wow, so we've got viruses that can hijack cells, replicate like crazy, and even hide out undetected.
And that's not all.
What? There's more.
Oh, yeah, there are even weirder things out there.
Okay, I am officially freaked out, but also intrigued. Tell me more.
Well, have you ever heard of viroids and prions?
Viroids and prions. Okay, those sound vaguely familiar,
Yeah.
but I definitely need a refresher.
They're like the minimalist artists of the pathogen world, stripping things down to the bare essentials and still managing to cause disease.
Minimalist but mighty. Okay, I'm all ears. Let's dive into this even weirder world.
All right, let's do it. So let's start with viroids.
Okay, viroids.
Imagine a tiny, like incredibly tiny loop of RNA.
Okay.
And that's it. That's a viroid.
No protein coat.
Nope, no protein coat or anything.
It's just RNA running wild.
Basically. And they mostly cause diseases in plants.
Huh, interesting. So how do they even cause disease if they don't have any of the machinery that viruses have?
That's a great question. And honestly, scientists are still trying to figure out all the details.
Really?
Yeah, but they think that viroids can basically mess with the plant's own RNA processing machinery.
Okay, so they're like disrupting the plant's internal communications.
Exactly. They interfere with gene expression, which can lead to all sorts of problems for the plant.
It's like a tiny saboteur sneaking into the plant's control room and just messing with all the settings.
Terrific analogy.
So we've got viruses that hijack cells and viroids that sabotage plants. What else is out there?
Well, we've also got virusoids.
Virusoids, okay, what are those?
They're kind of similar to viroids in that they're also tiny RNA molecules.
Okay.
But unlike viroids, they can't infect a cell on their own.
So they're even more minimalist than viroids.
Right. They need a helper virus to actually get inside a cell.
Oh, so they're like the ultimate tag-along pathogen.
Exactly. They hitch a ride with another virus, and then once they're inside, they can replicate and sometimes even make the symptoms caused by the helper virus worse.
Wow. So they're adding insult to injury.
That's one way to put it.
Okay, so we've covered viruses. Virusoids. Are you ready to move on to the really scary stuff?
Let's do it.
Okay, bring on the nightmares. What's next?
Prions.
Prions. Okay. Those sound familiar, but I don't remember much about them.
They're unlike anything else we've talked about so far. They're infectious proteins.
Wait a minute. A protein can be infectious.
Yep. No genetic material, just a misfolded protein that can cause other proteins to misfold as well.
Okay, that's creepy. How does that even work?
It's pretty wild. It's still an area of active research, but scientists think that when a prion comes into contact with a normal protein, it can basically force that normal protein to change its shape.
So like one bad apple spoiling the whole bunch.
Exactly. And then you get this chain reaction where more and more proteins become misfolded.
Then what happens then?
Well, these misfolded proteins, they can clump together and start disrupting the normal function of cells.
Oh, no.
And this can lead to some really nasty neurodegenerative diseases.
Like what?
Mad cow disease in cattle, for example, or Creutzfeldt-Jakob disease in humans.
I remember reading that prions are incredibly resistant to destruction.
Yeah, that's what makes them so scary. They can withstand heat chemicals, radiation, you name it.
Like they're indestructible zombies.
Pretty much. And sadly, there are currently no effective treatments for prion diseases.
So once you've got prions in your system, there's no way to get rid of them.
Not that we know of yet. The damage they cause is often irreversible and ultimately fatal.
So we've got viruses that can hijack cells, viroids that sabotage plants, and prions that spread through misfolded proteins, causing devastating diseases.
Ew.
It's kind of overwhelming to think about.
It is. It really highlights how even the smallest things can have a huge impact.
Absolutely. And it makes me appreciate the work that scientists are doing to try and understand these things.
Oh, for sure. The more we learn about them, the better equipped we'll be to protect ourselves and the world around us, you know?
Totally. And speaking of scientists, earlier we were talking about how they have to use all sorts of clever techniques to grow viruses in the lab.
Right.
Do those same techniques work for viroids and prions?
That's a great question. And it leads us to another layer of complexity in studying these things.
Okay, I'm intrigued. Tell me more.
Well, remember, viroids are basically just naked RNA. They don't have that protein coat that viruses use to attach to and enter cells.
Oh, right. So they can't just hijack a cell's machinery in the same way that a virus can.
Exactly. To study viroids, scientists often have to introduce the RNA directly into plant cells.
So how do they do that?
It can be tricky. Sometimes they use mechanical means, like physically injecting the RNA into the plant cells.
Oh, wow.
Yeah, or they might use a technique called agroinfiltration.
Agroinfiltration. That sounds intense.
It is kind of. Basically, they use bacteria to deliver the viroid RNA into the plant tissues.
So it's like they're bypassing the whole infection process and just sneaking the viroid RNA straight into the cell's command center.
Exactly. And then they can observe the effects on the plant, see if it develops any disease symptoms or if its growth is affected.
That makes sense. So it's a lot more complicated than just growing them in a petri dish like we talked about with viruses.
Yeah, definitely.
So what about prions? How do scientists study those sneaky, shape-shifting proteins?
Prions are a whole different ballgame. Well, remember, prions aren't like viruses or bacteria. They're not living organisms that can be easily grown in a lab.
Right. They're just misfolded versions of proteins that are already present in our bodies.
Exactly. So it's not as simple as just introducing a prion into a cell culture and watching it replicate.
That makes sense. So how do they do it?
Well, a lot of prion research involves using animal models.
Okay, like mice or something.
Yeah, mice, hamsters, things like that. They use animals that have been genetically modified to be susceptible to prion diseases.
So they can study how the disease progresses in a living organism.
Exactly. They can observe the effects on the brain and other tissues.
I can see how that would be really valuable for understanding these diseases.
Yeah.
But are there any alternatives to using animal models?
Yeah, that's a really important question, and scientists are always looking for ways to reduce or replace the need for animal testing.
Good.
One promising area is the development of cell-free systems.
Cell-free systems, what are those?
It's basically a way to study prion replication in a test tube.
Oh, interesting.
Using purified proteins and other cellular components.
So they're recreating the prions environment in a controlled setting without using any live animals.
Exactly. It's pretty amazing stuff.
It is. And are there any other techniques they use?
Yeah, there's also something called protein misfolding cyclic amplification, or PMCA.
PMCA, okay.
It's basically a way to amplify prions in the lab.
So they make more of them so they're easier to study.
Exactly. This helps scientists detect and study prions much more easily.
Wow. So it sounds like the study of these acellular pathogens, viruses, viroids, prions, is constantly evolving,
Absolutely.
with researchers pushing the boundaries of what's possible to unravel the secrets of these tiny but powerful entities.
And it's so important that they do.
I agree. It's a little scary to think about all these things that we can't even see causing so much trouble.
It is, but knowledge is power. The more we learn about these things, the better equipped we'll be to fight back.
Well, that brings us to the end of another deep dive.
Wow, that was a fascinating one, wasn't it?
It really was. We explored the world of acellular pathogens, those tiny but mighty entities that challenge our very definition of life. We dove into the intricate workings of viruses, those masters of cellular hijacking, and we uncovered the secrets of viroids, those minimalist saboteurs of the plant world. And then we ventured into the unsettling realm of prions, those shape-shifting proteins that can wreak havoc on the brain. It's a reminder that the microscopic world is full of wonder, complexity, and yes, sometimes a little bit of terror. But as always, the pursuit of knowledge is the best way to combat fear. So to our listeners, keep exploring, keep questioning, and never underestimate the power of the unseen world. Until next time, stay curious.